Cell wall of bacterial cell. Now we have learned about the basic classification of bacteria, in which they are basically divided into eubacteria and archaebacteria. So eubacteria, they are further divided into gram positive and gram negative and cell wall lacking bacteria. This means that the bacterial classification is based on the presence or absence of cell wall. There is a category of bacteria which possess cell wall, and there is a category which lacks cell wall. Absence of cell wall is there in case of Mycoplasma group of bacteria. The bacteria in which the cell wall is present are gram positive eubacteria and gram negative eubacteria. Likewise, archaebacteria are also divided into gram positive and gram negative archaebacteria. Now we'll talk about eubacterial cell wall structure. So eubacterial cell wall structure is of two types: the cell wall which is present in gram positive bacteria and cell wall present in gram negative bacteria. First, let us see the cell wall of gram positive bacteria. So in this figure you can see that this is cell membrane having phospholipid bilayer and presence of proteins, and outside to this cell membrane there is presence of this cell wall in gram positive bacteria. So generally we know that gram positive bacteria have high percentage of peptidoglycan. That means the thickness of the cell wall in case of gram positive bacteria is more as compared to gram negative. That means the cell wall of gram positive bacteria is thick because it has a thick layer of peptidoglycan. We can see that this cell wall of gram positive bacterium, it consists of peptidoglycan and teichoic acid. Sometimes there may be presence of lipoteichoic acid. So this peptidoglycan, it is made up with two amino sugars called as N-acetylmuramic acid, that is abbreviated as NAM, and N-acetylglucosamine, which is abbreviated as NAG. So this peptidoglycan is the it is composed of alternative units of NAG and NAM. Likewise, and these layers of NAG and NAM, these are cross-linked to another layer of NAG and NAM, which is further cross-linked to next layer of NAG and NAM, and this is how it leads to formation of several layers of peptidoglycan. Along with that, this layer contains teichoic acid, which is bound to the cell membrane, teichoic acid and lipoteichoic acid. So teichoic acid, it consists of, it is a polymer. It is a polymer of glycerol and ribitol phosphate, and if it contains lipid, then it becomes lipoteichoic acid. That is the structure of cell wall in case of gram positive bacteria. Let us see in detail the structure of peptidoglycan. The structure of peptidoglycan we have already learned in the chapter of biochemistry, the last chapter. So this peptidoglycan, as we can see, it consists of NAM and NAG. These are the amino sugars. So NAM is N-acetylmuramic acid and NAG is N-acetyl glucosamine. These are alternatively arranged like this. Further, this NAM, N-acetylmuramic acid, is attached to a short chain of amino acids. So you can see that there is a short chain of amino acids, usually 4 to 5 amino acids here and 4 to 5 amino acids here. And these short chains of amino acids which are present on NAM are further cross-linked. They are cross-linked through a chain of amino acid which is made up with 5 amino acids. So it is called as pentapeptide chain. So you can see in detail, this is the NAM of upper layer, this is the NAM of lower layer. Both are attached to a short chain of amino acid, and these two amino acid chains are cross-linked using this pentapeptide chain. This is the complete structure of peptidoglycan. Now, the structure of cell wall in gram-negative bacteria. So this is the cell membrane, similarly present as like that of present in gram-positive bacteria. External to the cell membrane, there is presence of a layer of peptidoglycan, and this peptidoglycan layer is thin as compared to the peptidoglycan layer which is present in gram-positive bacteria. This is thin. We have already seen the structure of peptidoglycan. Further, it is attached to a lipoprotein. So this lipoprotein is then further attached to one more membrane here, which is having structural similarity with that of this phospholipid bilayer, and that layer is called as outer membrane. So this space between cell membrane and outer membrane, this is called as periplasm, and this periplasm contains peptidoglycan. Now, what is this outer membrane? Outer membrane is made up with phospholipid bilayer. This is phospholipid, this is phospholipid. So these phospholipids are arranged in a bilayer, and this bilayer, it forms outer membrane majority. This bilayer, it also contains some transport proteins which are responsible for the transport of nutrients from outside to inside. Similarly, there are channels which are made up with proteins also. They are called as porins, which are also involved in the transport process. So this outer membrane, it consists of a phospholipid bilayer and one more component which is shown here, this one. This is called as lipopolysaccharide. This lipopolysaccharide is present in the phospholipid bilayer. What is this lipopolysaccharide? It consists of three polymers or units. This is lipid A. Then this lipid A is attached to a short chain of sugar that is called as core polysaccharide, and further it is attached to one more chain of monosaccharides which is called as O polysaccharide, and further it is also you can see here the presence of fatty acids. Now this lipopolysaccharide which contains this O polysaccharide, it is also called as O antigen, and this O antigen it consists of glucose units, galactose units, mannose units and rhamnose units. The structure of this O polysaccharide or O antigen is very important. It is useful for the classification of bacteria, and this site is also important for bacteriophages to attach to the cell wall. Now the functions of cell wall. The rigid cell wall of bacteria, it maintains or it gives a characteristic shape. That is the first function. Second, high pressure and other severe physical conditions. So this rigid cell wall it protects the cell from high pressure or other severe physical conditions. Third, cell wall serves as a barrier that prevents the entry of toxic chemicals or hydrolytic enzymes that may cause damage to the cells. For example, it prevents the entry of dyes, antibiotics, bile salts, heavy metals, enzymes, and this is how it prevents the entry of these chemicals, toxic chemicals inside the cell. The O antigen of LPS is lipopolysaccharide. It determines the antigen specificity of gram-negative bacteria. The matrix protein present in the outer membrane, it acts as a receptor sites for bacteriophages. Similar cell wall is also present in case of Archaebacteria. However, its composition is different. The archaebacterial cell wall it consists of proteins, glycoproteins and carbohydrates, that is polysaccharide. The peptidoglycan layer which is present in eubacterial cell wall, it is called as murine, whereas similar material which is present in our archaebacterial cell wall, it is called as pseudomurine. So pseudomurine is different from murine present in cell wall of eubacteria. Sometimes in some classes of bacteria, distinct type of cell walls are present. For example, cell wall of Mycobacterium. Mycobacterium cell wall it contains high amounts of lipids, mostly wax. These waxes along with glycolipids makes the cell wall of Mycobacterium very tough. So this presence of waxes in the cell wall of Mycobacterium, it gives an additional property to this bacterium. That means it makes that bacterium a distinctive staining characteristic. So because of this waxy material, these mycobacteria cannot be stained by simple staining procedures, but it requires a special staining procedure, and this staining procedure is called as acid fast staining.